microorganism. We also validated ourselves from urine for Mycoplasma genitalium for male, because when you want to test for male urethritis, then you want to do urine. In case of neonatal infections, then you would do the swab from neonate and also place it in e-swab and inform media. Or in case of amniotitis, then you could collect amniotic fluid. In case of meningitis, you could collect cerebrospinal fluid. In case of male infection, prostatic fluid, semen collection, and all that could be collected in sterile container and then extracted and done by molecular assay. And tissues also could be tested. Now, the testing for mycoplasma, as you know, and as Dr. Cole mentioned, that it's very difficult to really grow them, and it's very difficult to collect and transport. They need special conditions to maintain the viability, and they require special media and then special conditions to grow. It takes about 3 to 5 days to grow and to identify, but most favored one today is by molecular method, and will not take more than a couple hours if you use real-time PCR, or within a day to really detect and report. No serology currently for mycoplasma, only for mycoplasma pneumonia, but I'm not talking about that one right now. This is just the genital mycoplasma. In our lab, like I said, we offer as a profile and as a single test. So physicians can order just uroplasma, just mycoplasma homini, or just mycoplasma genitalium, or the whole together in a profile. So they just mark the whole panel, and we validate it from genital mycoplasma using from cervix or vaginal swab in M4 media, e-swab, and urine, like I said. And we use Infinity Urogen Quad assay, which is a profile developed by AutoGenomics, and it's performed on Infinity system, which is a fully automated multiplexing molecular diagnostic platform that uses novel assay-specific biofilm chip or microarray, chip array technology. It's very easy to be performed. We validated in our lab using Magnum Pure automated extraction system, and it takes about 60 minutes to really extract 24 specimens, and then about 85 minutes to do the PCR. We validated on 9700 ABI thermocycle, and then takes about 210 minutes to do the detection on the Infinity system. This system can work with other extractor system. They have validated themselves, or they have clients that they also use different extraction system, and also with the Eppendorf thermocycle. The timing is almost the same, and the flow is almost the same. In our study, we actually used 50 patient specimens and 17 spike specimens. What we did, we ordered a TCC infected cells or bacteria, and we spiked about 17 specimens with a different concentration, and you'll see ranging from 100,000 to 780 copies of infected cells with bacteria. Of those that we used just the patient specimens, we had 30 urines and 20 M4 media. And in comparison with a reference method, the correlation of uroplasma and urolyticum was 98%. 1 of 5 of 6 were positive, and we didn't have enough specimen to repeat in our lab to really confirm that discrepancy, but reference method had 6 positives, we had 5 positives. So that decreased the correlations to 98, and Mycoplasma homini and Mycoplasma genitalium really gave us a good correlation of 100%. So, and that we were not satisfied with the positive rates, because 9, 10 is not enough to really validate the assay. So we did, like I said, spiking with infected ATC cell lines or bacteria, and then we did a different concentration. And I'll show it to you using different concentrations, single plex, which means we tested every single one, and then we mixed them all together, because we wanted to show that can we detect. And as you may see, for when we used 12.5 nanograms, this one was equivocal. It was not positive, but at least we can see that all 3 together could, the competition for PCR will not inhibit. We will detect them all 3 in case they are all 3 present in the specimen. Also, because we validated from urine from males, we wanted to make sure that we don't miss and that the volume has no impact on the sensitivity. So what we did a 1 ml and 3 ml. 1 ml we extracted, and We spin down 3 ml and suspend it back in a 1 ml, and we extract it, and we realized that positive ones, 3 ml versus 1 ml, was not much of the difference, so we validated as a cutoff using 1 ml of urine instead of 3 ml. And in case that we get equivocal results, we could go back and then use 3 ml to make sure that we didn't miss anything. So yeah. Also, we did the reproducibility using a control DNA, and our techs, different days, they run 15 runs, and we use the mean. We use the relevant, RFUs, which is a relative fluorescent unit that they report on. So we monitor that by each run, and we got a pretty good mean. So it's very reproducible and reliable assay. Also, obviously, we had to do some specificity study. So what we did, we used a positive specimen with Mycoplasma genitalium. We spiked it with chlamydia, Neisseria, Trichomonas vaginalis, group B strep, and HSV. We test them for all those. We do that in our lab. They all were positive, and we again went there and tested for Mycoplasma genitalium, and we didn't see any cross-reactivity or any inhibition because of the presence of the DNA of these genital pathogens that usually are present in the specimens. So the specificity was the specificity of the probes and primers that we use for this assay are really specific and then don't cross-react with other genital pathogens present in the specimen. We also did the stability studies because we have clients, we do some clinical trials that they might not be able to send the specimen within 7 days. So we tested 1 day and 7 days at 4 degrees storing the specimen, and we didn't see that much of the difference really after 7 days. So we validated the stability at 4 degrees for 7 days. In summary, genital mycoplasma assay should be offered as a part of the STD testing for prevention of neonatal and adult-related diseases, as you saw that on a previous slide. It could be ordered as a screening test or diagnostic test, and the Infinity Urogen Quad assay performed on the Infinity system is a novel multiplex PCR assay used for detection of these 3 pathogens from patient specimens. Is an accurate, reliable, with a high sensitivity and specificity assay. And I would like to thank you to AutoGenomics R&D and technical support for supporting us on some testing. My team from the molecular lab and DCL Molecular Laboratory, Molecular Micro, and thanks to all of you for attending and your attention. Thank you.
My name is Bill Miller. I'm the CEO of AI Biotech. AI Biotech is an